Okay, welcome to week 3. So before I go into the PowerPoint and explain you the topic of the week, which is DNA microarrays, I would like to go over the material which I have posted in week 2 folder, and it is labeled as extra reading material. So if you see this document which I posted on week 2, so this is a document which says introns and axons. I'm going to discuss a few things in these two extra reading materials first, because these few concepts which I'm going to talk to you, they will be very useful to you in understanding this lecture, as well as when we reach to the NCBI website and when we'll be doing various computer algorithms. So you definitely need to know all these terms, and it should be very clear in your mind. So this is basically the structure of a gene, the DNA. I have already explained what are introns and what are axons. So once again, introns, so in the DNA, in the DNA where our genes are, these genes are made of DNA, which is a chemical. We have talked about the structure of DNA. So there is a lot of regions in the DNA which don't code for any proteins. As you know, DNA codes for proteins. There are a lot of regions in the DNA which don't code for any protein. So that region of DNA is called junk DNA, and that junk DNA has been given the name introns. And the regions of DNA, or the part of DNA which is coding for proteins, they are called axons. Okay, so there are two terms, introns and axons. Introns, they don't code for any protein, that's junk DNA, and axons, they are protein coding part and they code for the proteins. So this is our DNA. As you know, DNA is expressed into proteins, and how it is processed through two processes, transcription and translation. First of all, the DNA is going to be converted into mRNA. So this is about the DNA, and let's go to the second part. So this document says mature mRNA. So when the information in DNA, which is in the form of sequence of DNA, is converted into mRNA, first of all, it is mRNA is made, and that is all made on the basis of the sequence the DNA has. So everything, whatever the sequence in DNA, is going to be coded, and that will be coded on the basis of complementary base pairing rules. But the introns, which is the junk DNA, it is also coded. Before this mRNA can go into the ribosome and can make a protein, these introns need to be removed. And why do they need to be removed? They need to be removed because this is all junk. They don't code for the proteins. So basically, in a process, so first of all, the primary transcript is formed. That is not ready yet. It needs to be processed, and in that processing, introns, they are removed, and all the axons, they are joined together or they splice together, and this process is called splicing. So in the process of splicing, all the axons which are going to code for the protein, they are going to be joined, and the introns will be removed. And what is going to be formed? A structure like this is going to be formed. Okay, what is this? This is a mature form of mRNA. This is mature form of mRNA, and this mature form of mRNA is going to be going to the ribosome, and it is going to be translated. This form of mRNA is ready for the translation. I would like you to understand the structure of this mRNA, which is mature form. You can see here the ends of this mRNA, they are little modified. On the 3' end of this mRNA is attached a poly A tail. Now what is a poly A tail? It is a short stretch of A's, adenines, linked together, and they added here. So this is a tail, which means it's a short stretch of A's, and they are attached to the 3' end. And on the 5' end, it is called a 5' cap is attached. Basically what it is, 5' cap is a guanine, and you know what guanine is. It is modified and it is attached here. Now why this end and why this end, they are being modified? These two ends are being modified because mRNA has to move from nucleus has to go to cytoplasm, and RNA is very sensitive molecule, and it has to cross over the cytoplasm where there are a lot of enzymes. There are a lot of these hydrolytic enzymes are there. This mRNA can get degraded on the way. So to prevent that, the ends of the mRNA, they're modified so that it becomes more stable, because mRNA is the one which will be carrying the message of DNA to the ribosomes to prevent its degradation. And another thing, if you see here, this region is called CDS. It is called coding sequence. Now this term CDS will be very useful to you when you will doing the computer algorithms, because when we reach up to the NCBI and start working on the different things, you will be encountering a lot of terms. So CDS is one of the term you will see there. Now what does CDS basically means? CDS means the coding sequence. And how the coding sequence is formed? The coding sequence is formed by joining all the axons together. So all the introns are removed and all the axons, they are joined together and they form the CDS. And this coding sequence has here start codon and here is the stop codon. You know what the codons are. So AUG is the start codon, and there are 3 stop codons or the termination codons. There is one region here which is called 5' UTR, and on this side there is a region called 3' UTR. What are UTRs? They are called untranslated region, which means this is some part in the mRNA and this is some part in the mRNA which is not going to be translated, which means it's not going to make any protein, but it's still there. So scientists don't know why we still have this region. Maybe it is involved in some kind of regulation. So this is the structure of mature form of mRNA. Point to be noted, you should know this term CDS. By now you should know the terms introns, axons, CDS, and also one point I would like to tell you guys here. Sometimes when you will go to when we will go to the NCBI site, you will see that when you want to find out some DNA sequence of some gene, and when you will add the name of the gene in the window, you will get a lot of options. And in that, the best thing for you will be to find out mRNA sequence. And I hope you understand why you need to find out mRNA, because in the mRNA, all the introns, they have been removed. We are not interested in introns, because introns, they don't code for any protein. It's all junk DNA. So whenever you need to find the sequence of some gene, you will get a lot of options. Go and try to look for mRNA. That should be your choice. And another thing you will find when you will see the sequence of the mRNA, so remember that this is RNA, but you will see you don't find any U's there, but instead you have T's. So that is going to surprise you. Oh, I have taken out the sequence of mRNA, but why there are no U's? Because RNA does not have any thymine. But how does it has no U but it has thymine? The answer for this thing is, this is cDNA. Now what is cDNA? This mRNA can be converted to cDNA. What is cDNA? It is called complementary DNA by the help of an enzyme called reverse transcriptase. There is an enzyme called reverse transcriptase. So it should be very clear to you what is transcription. In transcription, from DNA, RNA is made. That is transcription. But RNA can also be converted into DNA, and that process is called reverse transcription. To convert RNA to DNA, there is a requirement of the enzyme called reverse transcriptase. It can convert back. So when you convert DNA to RNA, the information, the sequence in DNA acts as a template. But now when you convert mRNA to DNA, mRNA is going to act as a template. So which means that now this is mRNA, this is mRNA. Now if you are going to convert this mRNA, and that DNA will be cDNA, that is a complementary DNA. That can be done with the help of reverse transcriptase. In that case, this mRNA will be acting as a template. So in this mRNA, as you know that all the introns, they have been removed. So when you make cDNA from mRNA, that DNA does not have any introns. So that is why on NCBI site you will find in the mRNA sequence, you will find the T's. There will be no U's because that is a cDNA. That is actually the conversion of the mRNA sequence into the DNA. So few terms should be very clear to you. What is the intron? What is the exon? What is a cDNA? What is a complementary DNA? What is the structure of mature form of mRNA? And what is CDS? This is the coding sequence, because when you will go to NCBI, they will give you information. This is CDS, so you should know that this is the code, protein coding part. Okay, so this is all the information I wanted to give you. So let's go to today's topic, which is DNA microarrays. So we will be understanding in this lecture why do we use DNA microarrays, what is the use of that, and how it is done. So DNA microarrays. The important thing about DNA microarrays, it's a part of a high throughput technology. So what is a high throughput technology? So high throughput technology is large scale methods to purify, identify, and characterize DNA, RNA, proteins, and other molecules. These methods are usually automated, allowing rapid analysis of very large number of samples. So when you can analyze a lot of samples at the same time, that is your high throughput technology. And DNA microarray is one of them, because in DNA microarray, by this technology, you can do the analysis of lot of genes at the same time, more than 1,000 genes at the same time. So I'm going to show you some cartoons. It's basically just to tell you the pre-genomics era, how was it? At that time, the scientists used to analyze one gene at a time. So it was a slow process, and people were like studying one gene at a time. So now comes the era, which is your post-genomics era. You can see here, the scientists now can study a lot of genes at the same time, and that has made it has made the efficiency of learning about all these genes very high. So we can study a lot of genes at the same time. So basically, when we are like as I have told you, DNA makes our genes, but there is a lot of DNA which does not code for any proteins. That is all junk DNA, and they're called introns. So they are usually removed during the process when the DNA is getting converted into mRNA. So if we want to study the gene expression, okay, if we want to study about the expression of genes, we need to learn about we need to know the level of the mRNA. So if we know, so knowing mRNA expression levels tells a lot about the state of the cell. So if we have, suppose we have two samples, and we have one sample is from a normal person, another is from the cancer patient, and we want to know what is happening in a cancer patient, that he or she is getting this disease, which means that some of the genes are being expressed in a different way, right? So that is why this person is getting cancer. Some of the genes are being expressed in a different way. So as you know, DNA itself does not do anything. It expresses into proteins, and the proteins are the ones which are the real players. So basically cancer is a disease in which the cells, they start dividing abnormally, and the process of cell division is driven by a lot of proteins. So which proteins are getting expressed? Either they are expressing in higher amounts, or they are expressing in lower amounts as compared to the normal people. That is why people are getting this disease. So we want to study that. So to study which genes are getting expressed, the best thing to understand will be the mRNA in both the patients. If we can understand what is the level of expression of, if we can see the level of mRNA in both, means in one patient, for one gene maybe the mRNA level is high, or another for another gene mRNA level is less, then we can say which genes are getting expressed in a higher amount, expressed in a lower amount. To understand the gene expression, we need to learn the expression of the mRNA. So proteins do most of the work, They are dynamically created and destroyed. So are their mRNA blueprints. Different mRNA expressed at different times and places. Knowing mRNA expression levels tells a lot about the state of the cell. So suppose we want to know about 1000 or 2000 genes at the same time in the two samples. So we can make use of DNA microarrays. With the help of DNA microarrays, we can study the mRNA expression of maybe 1000 or more than that genes at the same time, and we can study the mRNA expression level of around 1000 or 2000 genes in the normal and the cancer patients. And by doing that, we can say which genes are getting expressed, or which genes are expressed in a higher amounts, which genes are expressed in a lower amounts. So we can use DNA microarrays as a diagnostic profile. We can use for cancer diagnosis. Also, not only for cancer situation, we can also do a lot of some very basic studies. Like suppose if you are trying to study some drug and you want to see how that drug affects the cell cycle of the cells. When you add some drug, how do cells start behaving? You can so basically whatever is the behavior of the cell is all on the basis of expression of the genes. And to study the expression of genes, we need to understand, or we need to study the expression of the mRNA. So these DNA microarrays, they can be used for various purposes. They can be used for diagnosis of cancer, not only cancer but various other diseases. All the diseases which are genetic, which are on the basis of the genes, they can be studied by DNA microarrays, or we can even make some basic studies. So this is your DNA microarray. So it's a snapshot that captures the activity pattern of thousands of genes at once. And what is basically a DNA microarray? They are based on the ability of complementary strands of DNA or DNA and RNA to hybridize to one another in solutions with high specificity. So this is basically your DNA. This is custom spotted array, and this is Affymetrix gene chip. What is a gene chip? Affymetrix is a company which makes these gene chips. So basically you can buy from this company. So basically or you can even, this is custom spotted arrays. I mean, you can buy them, or you can ask them to make these microarrays. Suppose you are interested in some specific genes, you can ask them. You can even create them custom. You can custom design them. So basically what is on the DNA microarray? There are some spots of the DNA. So as you know, the DNA, like the computer chips, small spots are there. So similarly here, all these spots, if you see, all these spots are representing the gene. Suppose I want to study 1000 genes. So I will ask the company, or even you can make in your lab, you can put the DNA for the various genes here. So every spot there is the DNA from the various genes, and then you can take your own sample and you can convert it into mRNA, and the you can extract mRNA. Basically you can extract the mRNA, and this week's lab is a virtual lab on microarrays that will make you this concepts very clear. It's a very good lab. So you can have your mRNA, you can convert it into cDNA, and you can just put your sample here, and on the basis of hybridization. So if some suppose here is one gene which you have spotted here, that gene is being expressed in higher amounts in cancer patient, it is going to go and bind here. Okay, and you will be labeling your genes with the different colors, and then on the basis of your colors, like you can label the DNA from normal patients with green color, from the cancer patients you can label with red color, and wherever this gene is more, it is going to show that color. Suppose here you can see red, maybe this sample, suppose if the from the cancer patient the sample is labeled with red color. So if you see red here, it means this gene is more expressed in the cancer patients. And if it is a green, maybe this gene is in normal people. If it is yellow, which means it is in both. So this is your DNA. This is the basics of microarrays. So DNA attached to the solid support, it can be glass, plastic or nylon. So this is the solid support on which the DNA is attached, and whatever DNA the genes are attached, these are either done by some companies. There are a lot of companies which make these microarrays. You can ask them, okay, I want to study these kind of genes. They can tell you, okay, you can buy this, or you can custom design them. You have some specific genes in your mind and you want to know what is happening in the patients, or you're doing some basic research, you want to see, you can even custom design them. So basically there are DNA attached to the solid support. Then you can label your RNA. So from your own sample you need to extract RNA, and you can basically convert it into DNA, which will be cDNA. When you convert RNA to DNA, it is cDNA, which is complementary DNA, and or even sometimes you can take directly RNA also. So RNA is labeled. You have to, if you have two kinds of samples, cancer patients and the normal, you need to label them with different fluorescent dyes so that you can later distinguish. So bound DNA is the probe. The DNA which has been attached to solid support, it is called probe, and the labeled RNA which you are going to put on this is a target. So I'm going to explain you this technique. You will be understanding very nicely. So suppose we have these two kinds of cells. This is cell type A, this is cell type B. Okay, what you have to do, you have already bought the DNA chip. You have this chip here. What you need to, because that's ready made. So what you need to do on your part, you will be extracting mRNA from your samples, from your cells. Then you are going to make the cDNA, complementary DNA from the mRNA, and you are going to label them with different fluorescent dyes. Suppose from cell A you are labeling with green, and with B you are labeling with red color. And then you have to add your samples on the DNA microarray one by one, and then on the basis of hybridization, because DNA can hybridize with DNA, DNA can hybridize also with RNA. So on the basis of when you add your sample on this chip, hybridization will take place, and it all depends which gene is being expressed more in which sample. Suppose it is more, some gene is more in the cell type B, you will see the red spots, and whichever gene is more in cell type A, you will see the green spots. So and so you can see here, if this is a gene which is getting green color, so it is more in A. If it is red, it is more in B. If you see yellow, it is equal in both. So as I said, uses of microarrays, you can study, it can be used for diagnosis purposes. It can also be used for studying some basic mechanisms. You can do tissue profiles. You can check which genes are being so it has a lot of promise in medicine, especially cancer research. So this is just data for the you can use the DNA chip to understand which genes you can study the expression of various genes in the leukemia patients. So what genes are behaving differently between ALL and AML? These are two kinds of your leukemias. And so it has potential uses in diagnosis, prognosis, and the treatment. So it basically explains you the technology. So basically with the first of all, you can purify the DNA, and with the robotic printing, these DNA, you see these spots, this can be attached on the on this chip, and then you can make your DNA. You can extract your mRNA, and that mRNA can be converted into cDNA and labeled, and then you can pour it, and this is what it is basically. So each feature or spot represents a specific expressed gene. The fluorescent intensity of each feature correlates with the expression level of the gene in the samples under investigation. So basically, how does it work? It works on the hybridization principle. So this is your DNA. So as you know, the two strands of DNA, they're linked with each other by the hydrogen bonds, and probe is it's a single-stranded DNA which is attached on the on this your chip. It can be glass, plastic. It can be glass or plastic. So probe is DNA spotted attached to the solid substrate. So you can see here, this is just attached on this support. And now what is your target is the fluorescent labeled cDNA representation of the mRNA, and is hybridized to the probe. So you can have these your target, you can have two kinds. Basically, you can have sample from normal patient, you can have from cancer, or it can be some journal study where you are treating your tissues with some drug. You can label them differently. You can extract RNA from them, convert it into DNA, and you can then pour your cDNAs on the chip, and you can see according to whatever gene is more in whatever in the normal situation or in the cancer or drug treatment, that gene is going to go and hybridize more there, and on the basis of that you will get the different colors. And then after you are done with this, there are laser scanners. You can add your chip into that and it can read for you. So this is all what your microarray technology is, and this week's lab is all about this, where you will actually do the experiment and you will really understand that.